So this is part 4. This section is going to be slightly more complicated. There's going to be a lot of different enzymes you have to learn. So, paper and pen, write it down, however you want to do it. I'm just going to go over the image again. So at this stage, the food has come in. It's going down in the form of a bolus. It's opened up the cardiac sphincter and it is in the stomach. We're going to look at the function of various accessory organs, the liver, the pancreas that you can see right here. It's kind of a almost like a leaf looking thing that goes, you kind of see the outline right there. Pancreas, and then we're gonna talk about what the gallbladder does, and that kind of stuff. And pay attention, this portion here has, right there is going to be your pyloric sphincter. The pyloric sphincter is not as tough or hard as the cardiac sphincter, because the job of the cardiac sphincter is to make sure that the acid doesn't back up into the esophagus. The pyloric sphincter, on the other hand, it like allows the acid to go, the chyme to go down, and then it's neutralized. So this, excuse me, so this one's not as tough as this one. Okay. So so we're going to break down basically the 4 different food groups, right? Carbohydrates, proteins, nucleic acids, and fats. So we're going to look at each one of them. This is going to kind of tie into cellular respiration a little bit. Again, one of you has done cellular respiration and the other one hasn't, so don't worry about that. I'm not going to ask you any questions on cellular respiration here. Step 1 is in the mouth. There's mechanical digestion. It breaks down the food. Carbohydrates, you're going to see long chains of carbohydrates can be divided into monosaccharides, polysaccharides, and disaccharides. Monosaccharides would be something like glucose. Glucose is a monosaccharide, it's just one unit, C6H12O6. So that's one unit. You have disaccharides, something like lactose. Lactose is made up of glucose and galactose. You have maltose. Maltose is glucose plus glucose. Sucrose, which is table sugar, which is basically glucose plus fructose. And then you have something called as polysaccharides. Polysaccharides meaning there's many sugars. Something like starch, which you can only find in plant products. And then you have glycogen, which is only found in animal products. These are just long chains of glucose molecules. In the mouth, the food is mixed with salivary amylase. Amylase is the enzyme. It's mixed in with saliva. A lot of it is water, and then you have the amylase. It kind of starts breaking down the material into polysaccharides, smaller polysaccharides. So let's assume they were 100 glucose molecules. It breaks it down into manageable 20s, so 520s. Okay. From there, it goes into the stomach. The stomach has a very acidic environment, so it basically neutralizes the amylase. The amylase is denatured, so it is not doing, there is no carbohydrate digestion in the stomach. The carbohydrates do not break down in the stomach. From there, from the stomach, again, it goes back into this little part called the duodenum. In this duodenum area, you're going to see most of carbohydrate digestion takes place. The acid, the stomach has a lot of acid. So once it comes into the duodenum, the pancreas pumps in a bicarbonate. Most of the enzymes that the pancreas pumps in. Now remember, these enzymes that they're pumping in, they're all water soluble. They're like dissolved in water. So it is very alkaline. It is very basic in nature, so it kind of neutralizes the acid. The chyme is at a pH of 3 or 4, and so it neutralizes it by pumping in bicarbonate. Neutralizes it, brings it down to like a manageable 7.4. At that stage, the amylase is again activated. Once the amylase is between 5 and 7.4. the amylase is activated, and then it breaks it down further. So then the polysaccharides are broken down into disaccharides, that means two sugars, and as it goes through the small intestine, those disaccharides are broken down, are broken down into monosaccharides, single sugars, glucose. It's glucose that's actually absorbed into the system. Galactose is a part of glucose. It's an isomer of glucose. It's just a different shape where the OH is organized. So carbohydrate digestion is pretty straightforward. Step one, in the mouth, it mixes up with the salivary amylase. It's broken down into smaller polysaccharides and maltose, which is sugar, which is glucose plus glucose. It's not that. So maltose is broken. It's broken down into maltose, and then it goes down, and in the in the duodenum section, once the stomach acid is neutralized by the bicarbonates in the pancreatic enzymes, it's then broken down into disaccharides. Then it's further broken down from the same amylase enzymes in the small intestines into monosaccharides, and then these monosaccharides are absorbed into the bloodstream. The inside of your small intestine has this finger-like projections. It almost looks like when you watch those underwater videos, you see those tube worms. It looks like that. So they're like these finger-like projections, and they're called microvilli. They're made out of a very simple epithelial tissue, so they allow for diffusion based on concentration. Along the concentration gradient, they allow for smaller molecules like monosaccharides to pass into the bloodstream. That's carbohydrates. Proteins are a little more complicated. Protein digestion, again, a mechanical digestion. So it breaks down into smaller pieces in your mouth. And then there is no digestion of proteins happening in the mouth. Remember, the word digestion means that it is breaking down these macromolecules into their individual monomers. That is, the word digestion just means that you're taking something that's a large molecule and breaking them down into smaller components, which are essentially their monomers. So there's no digestion of proteins that happens in the mouth. However, it is mechanically broken down into smaller pieces. Again, the stomach is not trying to digest a whole piece, a 72 ounce of steak. Once it reaches the stomach, if you remember in the previous video, we talked about this, pepsinogen is activated by the hydrochloric acid. It becomes pepsin and then breaks down into smaller polypeptides. So let's assume that you have a protein that is 400 amino acids long. So it's breaking down into 50 amino acids, 850 amino acid smaller polypeptides. If you remember in the previous video, I talked about it. You have one amino acid and the other amino acid is connected by a bond, and that bond is covalent bond. This covalent bond is called as a peptide bond. Once it enters into the duodenum, again, this whole section here is everything is in the duodenum. Once it enters into the duodenum, the pancreas, again, along with that pancreatic amylase, it's also pumping in trypsin and chymotrypsin. So it breaks it down further into smaller polypeptides. Then there's a pancreatic carboxylic peptidase. Anytime you see the word ASE, it is an enzyme. So it breaks it down further, and then it goes into a peptidase, which is two peptides, carboxylic, same thing, aminopeptidase. After a set of all of this, it breaks down into its amino acid, goes through the microvilli and into the bloodstream. So just to recap protein digestion again, there's no protein digestion in the mouth. There's only a mechanical digestion where it breaks it down into smaller pieces. In the stomach, pepsinogen is activated by pepsin, then combines, becomes pepsin, breaks it down into smaller peptides, goes into the duodenum. You have chymotrypsin and trypsin that breaks it down further. Then you have carboxylic peptidase that breaks it down further. Then it goes into the small intestine, the lining of the small intestine. It breaks it down further using dipeptidase and aminopeptidase. And at that stage, at the end of that, you see amino acids, single amino acids, and those amino acids cross through the microvilli, go into the bloodstream. So at this stage, here is where you're actually seeing some kind of absorption. There is no absorption anywhere here. Disaccharides are not absorbed, it's monosaccharides that are absorbed. A dipeptide is not absorbed, an amino acid is absorbed. And then we go into nucleic acid digestion. As you all know, nucleic acids, DNA and RNA. DNA has a sugar molecule. Both DNA and RNA have a sugar molecule, ribose. DNA has one less oxygen than RNA, hence deoxyribose. There are 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. And they also have a phosphate group. Remember that the phosphate group is very, very vital because it has to be used. It's used later on in the form in ATP and GTP and all of these other things. So it's broken down by, again, in the duodenum. No digestion in the mouth, no digestion in the stomach. It's just mechanical in the mouth, goes into the duodenum, and in the duodenum, a nucleases basically breaks it down into their individual nucleotides. Then those nucleotides are broken down by further into their nucleosides. The monomer of that is the 4 nitrogenous bases. You have a sugar and a phosphate. All of these are absorbed across the microvilli. Fat digestion, same thing. You do not have any fat digestion in the mouth and in the stomach. As you come into the duodenum, pancreatic lipase is secreted. Now, there's a couple of things that has to happen before a lipase can do its work. Lipids are not water soluble. So there is an enzyme, there is something called bile that is secreted. The liver actually secretes bile. Bile is stored in the gallbladder. If you have surgery where they take out your gallbladder, they actually have to, you have to kind of limit the amount of fat you eat. Step 1, before lipase can actually work on the fatty acids or on the fats, is to actually emulsify it. And bile acts as an emulsifier. I mean, think about it this way. If you're trying, it's like making salad, making mayonnaise, right? You have to put the egg in so that you can combine. So everything, the oil molecules and the water molecules kind of surround the egg protein. So they're all in one place. That's an emulsification. So bile does that. Once bile comes in, again, that comes into the duodenum. Bile kind of emulsifies the fat molecules. Lipase comes in and breaks it down. A fat molecule is basically a 3-carbon sugar. No, I'm sorry, 3-carbon alcohol, which is glycerol. This one. You have a, so I had to take a break there because my dog started barking. Once the emulsification is done, then the lipase comes in and breaks it up. The way it breaks it up is that it breaks it along the alcohol molecule. So think about it, vertically there is an alcohol molecule. There are 3 carbons in it, and it's a 3-carbon alcohol molecule. And horizontally there's fatty acids. Fatty acids basically have a lot of CHs, and on the terminal and on one of the terminal end where it joins with the alcohol, it has a COOH. So it breaks it down into the fatty acids, 3 fatty acid chains, and the glycerol chain. And then there are monoglycerides that are formed in that. Again, those are the ones, this is the one that's going to be absorbed. I'm assuming this is clear. If it's not, I think this picture is in your textbook. Kind of go over it in the textbook, okay? Most of the absorption happens in the small intestine. In the large intestine, most of the water is actually absorbed in the large intestine. That is why you have some constipation and diarrhea, and we'll get to that in a little while. The first portion, that first bend is the duodenum, and then it's in the duodenum that most of the digestion takes place. Most of your digestion is taking place in this area. Almost all of your nutrient absorption is taking place through the jejunum and the ileum, the little folds and crevices, folds of the small intestine. The small intestine is pretty large. It has these microvilli, which are epithelial tissue. It transports these